Hi, everyone. If I haven't already met you, I'm Veronica. Today, I'm going to be going over kind of two topics. The first is going to be going through some of the standards that we use in computational systems biology, and also talking a little bit about distribution using those standards. And then I'll also go through, give like a general talk about why reproducibility is important and some of the best practices for ensuring reproducibility in your modeling studies. And also during that, we'll go through a biomodeling workflow and just look at some of the tips implemented in practice. I'm working out of this day 4 session 1 folder, and the files we'll be working with right now, I'm just going to be using this reproducible distribution collaboratory notebook, and then later I'll kind of go through a PowerPoint presentation and use this very long biomodel ID name modeling study notebook. And for now, we'll just start with this one, and we'll have like a break in between and everything, but feel free to ask questions if any questions come up. Okay, so first topic is distributing standardized combine archives with using Tellurium. And you've seen some of the standards, I think, have been discussed a little bit throughout the course, but today we're going to be talking about primarily 3 standards, which are SBML, SetML, and then the Combine or OMEX archive. Okay, I'll just kind of get this notebook started. So we're going to start by just talking about some of the important export capabilities that Tellurium has. Basically, Lucian kind of demonstrated how models can be imported from the Biomodels database, given that you have, given you have a biomodel ID, and you can basically use a standard URL format to programmatically access your model of interest. And we're going to be using, again, the Jana Wolf model as an example of doing this, and we'll check out some of these standards. Right now, we're just going to be importing Tellurium and then also PhrasedML, and I'll talk a little bit about what that package does. Technically, it gets installed as well with Tellurium. Okay, so first, let's talk about SBML, which I think is probably the standard that's been brought up the most throughout the course of this summer school session. What actually is SBML? I think Janice described it a little bit, but it's a software data format that we use for describing models in biology. It's a little bit like HTML, but instead of for creating web pages, it's for creating models. And markup languages tend to allow you to be able to separate annotations of the information and also documentation about the content from the content itself, which uses these kind of standardized tags. And SBML in particular, it's independent of any particular software tool and not limited to any particular field in the life sciences, which means it's broadly applicable to the biomodeling domain. It's also open and free and very widely supported by a range of software tools, some of which you've already heard described. And there are libraries also, I think we've discussed libSBML a little bit, that help developers support SBML. Essentially, the goal there is to make software that's able to interpret tags in the standard and then encode your model so that it can be used in the tool. And these tools might allow for writing the model, simulating the model, visualizing a network, among a range of other tasks you might perform while you're modeling. So while the core features of SBML support reaction and process models, there are also some extensions that do support things like spatial models or constraint-based models, rule-based models, and logical models, which I think some folks have brought up. Yep, that's kind of a general overview of SBML. So we can talk a little bit about how you use it. I know you've seen a couple examples so far. As I think Janice said, you wouldn't want to write SBML yourself. The software you're using has to do that for you. You can basically use some tools. I should have probably linked in here, but there's some tools. There are sort of web pages like the SBML showcase that can help you actually find SBML compatible software. And then also, as we've talked about, the Biomodles Database is a good resource to look for published models that will be encoded in SBML. Okay, so let's do this quick exercise with the wolf yeast respiratory oscillations model that we've already used a little bit in the course. Basically, Tellurium allows us to import SBML models directly from the Biomodels database using a URL that's specific for that model. So here we're loading to this object name wolf. We're using te.load SBML model and then passing in this URL, which will link to a download for the Jenna wolf model, which is this biomodel 90. We're then going to run a simulation and go ahead and plot. I know it's already codes, the cells are executed currently, but so we've seen this a little bit before. This is all things you've learned, but it's really useful to be able to directly load these SBML models from the Biomodels database. And these are also curated models, as Lucian talked about. Many of them are, so it can be good to use to build upon if you want to in your own modeling study. It is the same as TE load S. I don't believe there's a difference. Is that correct, Herbert or Lucian?
Yeah, that's correct. It's just
Okay.
a synonym, I guess.
Yeah, okay. Okay, so in addition to import, we can also export SBML or antimony models, both from Tellurium, which we have probably discussed a little bit. So you can, now that you have this model loaded to wolf, to the name wolf, we can reset the model and basically do this export to SBML command, providing it a name for your model, .xml, just telling it that it's going to be in this SBML format. Current version essentially just means that whatever manipulations you perform to your model, and since you loaded it, you're going to export that. You can also export it from the origin, so when you provided the original version of the model, if you want to. Okay, so another option would be to potentially export the model to an antimony string instead, if you want to, if you're looking for, a modeling format that's going to be easier to read and edit. So I think we've seen this model.getCurrent antimony. This is I'm just printing out the antimony version of the model we just loaded. So perhaps I want to take a model directly from the Biomodels database that might be of a topic of interest, and then I actually want to be able to interpret it. So then I can go ahead and print the antimony version and get a better idea of what's going on in the model. So that's what's happening in this code block. This model is really well annotated, which we'll talk about in the second half of the session. Pretty much everything you're seeing here are standardized identifiers, and so we'll get into that a little bit more later. Okay, so now that you have your model and you've loaded it from the Biomodels database and converted it to antimony, this is just one way to edit the antimony model. So I just copied and pasted basically the printed antimony model from above into this wolf, gets te.load. So I have now my antimony model, and I can edit it in this code block if I want to, and we can go ahead and perform any manipulation we want to, and then we can export our modified version of the model here, either to antimony or to SBML, depending on the format you want to save. Okay, so I think you had the most background on SBML, but now we'll kind of get into a little bit of new territory, I think. First, we're going to basically talk a little bit more about the simulation standard, which is SbML. SbML encodes in a computer readable exchange format, just like SBML, the information that's required by these minimal information standards about the simulation experiment, whatever is necessary to enable reproduction of that simulation experiment. And it's been developed by this community as a sort of community project, and is defined in a detailed technical specification that also provides an XML schema just like SBML. So very similar, but instead of using this for model construction, we're using it for model simulation. It covers the description of kind of the most frequent types of simulation experiments that you'll see in the area. Obviously with standards, you don't have unlimited flexibility in how many different types of simulation experiments you could potentially encode or provide support for. But it largely is used for time course simulations and can also be used to specify what experimental data you want to use in your experiment, what models you want to use in your experiment, modifications you would apply on your models before you use them, which simulation procedures you would want to run on your model, what analyses and results you want to output, and then how your results should also be presented. So some graphical information. So we'll get a little bit into that. So for the purpose of demonstrating the use of SetML and also PhrasedML, which I'll talk about in a second, we're going to focus on just using this species here, which has KEBI ID 15379. This is Dioxygen going forward. So just pulling out one species in the model for the ease of showing you how to write a SetML experiment. Dioxygen, I just chose it. It's a substance that removes electrons from another reactant in a redox reaction. And since this is a metabolite of Saccharomyces cerevisiae, we can kind of get an idea of how this might come into play in a model of respiratory oscillations. So here I'm just running a simulation with, just like you've learned before. So this would be the model.simulate command, specifying the start time, stop time, number of time points, and the species that you want to record in your simulation results, and then plotting that here. So here's just the time course of this oxy species. So that it's really easy to write simulation experiments with Tellurium. If you want to then export to a standardized version of the same experiment, so if you want to export SetML, you would want to use this package in Tellurium at least called PhrasedML, which is a language and a library that provide a kind of text-based way to read, summarize, and create SetML files. Tellurium has functions that allow you to use PhrasedML as part of this larger system to create a model with antimony, describe the experiment with PhrasedML, and then perform the experiment. So in general, the PhrasedML language follows the SetML format, basically defines the models, the simulations, tasks, and then the output, and also can specify repeated tasks if you want. So we can kind of get into the anatomy of the PhrasedML string, essentially. So SetML models are always going to be models that are found with a URI. Typically, it's a relative path name for your file that you're specifying, or as modifications of some previously defined model. In PhrasedML, they're going to be defined using this syntax here, where you have the model ID gets model, and then the source SBML file. And if you have changes, then you would specify with and what changes you want to include. So here again, model ID, model, and then providing your source model, which here for me is going to be this wolf.xml that I exported above using the te.export to SBML. Okay, sorry, I should just keep the whole thing on the screen. Then a SetML simulation that is going to be basically a description of the type of experiment you want to perform. And if it was only going to describe the experiment itself, not the model on which it's to be performed, that's actually the job of the task. So we'll talk a little bit about that. Here, we want to perform a deterministic simulation. Basically, we're going to try to mimic this same simulation that we performed using Tellurium. So, you can do multiple types of simulation. Like I said, time course is pretty common, but you can also do steady state. For time courses, you can do deterministic or stochastic simulations. Here, I'm going to be doing this deterministic simulation gets simulate command uniform from 0 to 500 with 1,000 time points. And so the uniform here is indicating that we're doing a deterministic time course simulation. And this is kind of just telling you what each of those things are. This is, you're naming some simulation ID, you have the simulate command, and then simulate the type of simulation you want to perform. Okay, so after you have your experiment described, which is in this line, you're going to want to have your task defined. So a basic task is basically links the model to the simulation, and what it's telling you to do is run this simulation on that model. So in this case, we want to run the deterministic det underscore sim ID on the model wolf. So it's using these two, the model ID and the sim ID that we defined here. Okay, and finally, the last line in the phrase.ml here is to define the output. So here we're just asking it to do a simple plot. We're titling it with Wolf et al. Dynamics and the model ID, and then we want to plot the time species versus the oxy species. Sorry. And then, yep, so this is just describing, so the plot command, providing the title name, and then X versus Y. Okay, so that pretty much gets into the how to write a simple phrase.ml string. And then basically the next step is to actually generate the setML using this phrase.ml string. So to do that, we're gonna just reset our model. We're gonna get the SBML string because we're gonna need the SBML string to create our setML. And then the command is phrase.ml dot set reference to SBML. We're giving it the model that we want to use as our SBML reference. Again, remember we have a source model that's defined in the phrase.ml string, and then we're giving it this SBML that we just generated here. And then to actually get the setML, the command is phrase.ml dot convert string and passing in the phrase.ml string that we defined here. And so this is just going to print the setML that's defined. It's going to be similarly unreadable, kind of like SBML, but just to give you an idea of what the setML actually looks like, it is here. So here, I guess I'll just ask if there's any questions about the way setML experiments are defined, because I know it's kind of new.
I have a question.
Okay.
So the experiment that you have defined seems to be very structured. So if we have like if we don't write it this way, are we gonna get an error for the conversion?
So is there a particular line you mean? Do you mean this determinist that?
Yeah, like it looks very, yeah, it looks like an actual like line of code. Like this is assigned to that sim, or this is assigned to like, well, can we just like write things without like having a structure?
No, so it does have to be formatted. Like it does have to have your ID gets simulate command and then the simulation type you want to define, which there's a subset. There's in the phrase.ml documentation, there will be just a subset of possible tasks, or sorry, experiments that you can actually write here.
Oh, I see. And the variables should match the variables in your experiment.
Do you mean here, the time versus oxygen?
Yes.
Yeah, so those will have to be species that actually exist in the SBML model.
I see. Okay, thank you.
Mm-hmm.
The basic idea behind phrase.ml is to do for setML what antimony does for SBML.
Mm-hmm.
So yeah, it has structure. It's designed to be pretty human readable and human writable, but it does have a definite structure, and there are rules of their grammar lines and stuff like that. Just like an antimony, you can't quite write anything you want in antimony. You have to follow certain structures.
I see. Thank you.
Sure.
Okay, cool. So moving on to actually visualizing that and also exporting it. To export your setML, file, you can do tellurium.save to file, provide the Sbml name, and then also provide the Sbml string that you've just generated, which is saved again here with the phraseml.convert to string command. Okay, so once you've saved the experiment, I'm gonna then load and actually run the Sbml script. So here you would do tellurium.execute Sbml, and I'm providing it the path to the relative path to the Sbml that I'm just saving right before that. So we would hope that the outcome looks somewhat, it has the same dynamics as our former simulation. So here we obviously sampled for a longer time course, but you're seeing the oscillatory dynamics of this oxy species, as we were able to also visualize with the tellurium script. So the nice thing about this then, and about standards in general, is once I have this, I can export it to several other simulation engines, and they can regenerate these exact same dynamics, the exact same simulation experiment with a different simulator. So we'll get into that in a little bit. I'll show an example of doing that. Okay, so we've talked about SBML, which is the standard for constructing your model. We've talked about Sbml, which is the standard for simulating your model. And when you take the two and kind of put them together, along with potentially other files that may include data that's needed in your modeling workflow, you can generate a combine, what's called a combine archive. So these archives are a package that contains these related standards like SBML models, Sbml simulations. They join them together so that they can be easily exchanged between software tools. Tellurium provides the inline OMAX format for editing the contents of combine archives in a human readable format. So I'll give an example of that. And you can kind of use this function, convert combine archive, to convert a combine archive on disk to an inline OMAX string. And then you can also use this function execute inline OMAX that you'll see to execute the inline OMAX string also in your code. So that's basically what these two lines are doing here. You're going to first read your antimony model into a string. So here you have, again, naming it wolf antimony t, read from file, and then providing the relative string that we generated earlier. And then you're going to create this inline OMAX string, which is done by joining these two strings that we have here, wolf underscore antimony and wolf underscore phraseml. So it's joining together the antimony and the phraseml, and this is going to allow us to write the OMAX format, which again should be a sort of human readable version of the OMAX of what will be contained in the combine archive, which when you go ahead and print that string, you're going to first get the SBML, first get the model description, which is an antimony. You're pretty familiar with this at this point. And then afterwards, you're going to get, after all of this data, a very short Sbml experiment that we just wrote. So it should all look just like the string we wrote above. It's all the same. Okay, so now that we have this inline OMAX format, this human readable string, we can export it to the standard version, which is the combine archive. And again, that code is te.export inline OMAX, and providing it this inline OMAX format, naming it wolf.omax. And hopefully this will then export your combine archive. And with that, you pretty much have everything self-contained in this archive that you can distribute across platforms. Which we can try to, I can try to show you an example of running that in a different simulator. We'll see how this goes. So this simulator is Copazi. Maybe some of you have used it. It's one of the most common simulators, I think, in the systems biology domain. I'm just gonna import the combine archive, and I just previously stored it on my desktop, so it would be easy to access. Oh no. Okay, so I might not be able to do it with this model, which means I probably have a bug in my code somewhere. But I have an example using it later with a non-buggy version, so I can I'll show you the transitioning between different simulators a little bit later. Apologies for that. Yeah, load S is the same as load SBML model. It's just a shorthand for it, makes it a little easier to type. Yeah, in the meantime, are there any other questions that I can answer about any of these standardized formats for any of the code?
So one thing I'll point out is just that all the simulation that we've been doing so far have been using like Roadrunner commands, like r.simulate or something. So in order to use setml and phrasedml, you have to do that same thing a different way. We're trying to come up with a way that you could sort of do both at once and just sort of make it easy it would save the setml in the background and then let you export it. But right now, if you want to be able to take your experiment and move it around between different simulators or save it, publish it with your model, that sort of thing, then you'll have to like come up with the phrasedml or the setml version of it, of your experiment.
What was the bug Copazi showed?
Let's
I wonder
read
if you
generated.
have
I
an
think it
old
complained about
copy
not
of
being able to
Copazi,
find
maybe.
the model. Like, was the model not included in the Combine archive?
It should be. I mean, it was exported with this line, which should just
Yeah, we
export
should save it.
this full. Yep. I'll debug. It might be a naming issue somewhere,
Oh, yeah.
potentially. But
Yeah.
let me
I mean, it could be
regenerate.
because Copazi's old. Maybe you have an old copy of Copazi.
Well, so
Oh,
just
scroll
to
up
show
in your model real quick in the antimony thing. Sorry, I wrote. Like at the very top, I want to see what the model is called, right? I'm just called Wolf. Okay. So that I think it's automatically changed to wolf.xml. I don't know. There is some wolf. There is some naming magic that goes on in the background. Sometimes it's hard to figure out.
well, just so nobody loses too much hope, I guess I'll load another one that I'll show you the export for in the reproducibility portion. And so there are some warnings, mostly about units, and it's thankfully not going to prevent me from actually running the simulation. But so I just loaded in an omics file, and let's see if we can. So in Copazi, this is like a nice GUI that you can use to try to run your simulations. In plots, I can see, okay, you're gonna see in the next session that I'll be working with the repressolator, but here I'm just showing there's a plot that exists once I load this Combine archive in that's called repressolator dynamics, and we can see that this seems to be some code that will show a simulation, a time course simulation of some sort. And if I then go to time course and I go ahead and just run, basically I'm going to get this simulation that is predetermined, and you'll see in the example I show, it's called, basically the experiment is run deterministic simulation. It's running it on this, just plotting this protein PX. So you'll have more context for that later, but you can take your Combine archive, granted, if everything is formatted correctly, and should be able to export and then import into another simulator, which is really useful.
And is it true that biomodels are moving over to Combine archives, or have they? Lucian, maybe?
I mean, sure. So that's partly our effort.
Yeah, right.
is to get SbML files that actually work, but you can also so when you go to Biomodels, the website, you can see all the files and stuff, but you can also, at least through their interface, to the REST interface. I think you can ask for the an OMX file, and it will zip up everything and give that to you
Okay.
as an OMX file. So yeah, so another thing that we are working on is working with the Biomodels Database to get their SbML files up to par to actually produce the figures that they were originally curated to produce. So Biomodels has been around for a long time. Originally, they often would just have like a Copazi file and like use a model to simulate. They would create a figure, say that's great, and then they would publish the model. But if you want to reproduce the figure, then you would not know specifically, you could probably guess often, but not always, how to actually produce that figure, because you would have to simulate for a certain amount of time on a certain scale, asking for certain outputs. So Copazi now exports to SbML. So if you can find the old Copazi file, you can like load it and then export that to SbML, and then you can move that around. So we've been trying to use that and other tricks to sort of figure out how to get SbML out of those. And if we don't have anything, then we say like, well, you know, simulate for 10 seconds and output all the variables, and you can your data is in there somewhere, hopefully. So like as time goes on, hopefully we can like update and improve all the old Biomodels models in there.
The thing is, I mean, the hope is that the world will move towards Combine archives as a way of storing models, because the archive can contain, you know, all sorts of things, not just the SBML, but data, PDF files, etc. So it becomes a complete package that represents a model.
So as one example, you can do, as things move forward, you can now do parameter estimation. Parameter estimation involves actually collected data. So if you have a Combine archive, you can have your model, you can have your parameter data, you can have the SbML that describes how to do your parameter estimation, and then the results also. And then you could pull that into a different, stupid phone, a different simulator and run the same thing, and it would all work.
Yeah, there's a question from Michael about Copazi. Basically, what are its use cases, and does it have capabilities beyond Tellurium? I would say historically it's been used primarily as a GUI, which could be useful for people that maybe don't have as much programming experience, though Tellurium is relatively quick on the upstart with programming as well. It does have features that Tellurium maybe doesn't have implemented yet, but may have plans to. For example, I think for parameter estimation, does it have a Bayesian inference version of that?
That's where SBstoat things like SBstoat comes in.
Mm.
But in the Python world, though, you've got a lot more choices, though. I mean, somebody, was it yesterday or the day before, mentioned PyPesto. So there are other people writing parameter fitting packages for Python that can work with Roadrunner. So and SBstoat is a good teaching package, and it's also good for doing small models. But if you want to do large models, you should probably switch to one of the other Python packages. But it's Copazi and Roadrunner are pretty similar in capabilities.
I think Copazi has a lot of features. I would say it's pretty self-contained if you want to do a lot of modeling.
I mean, one of its claim to fame, I guess, is that it has a lot of built-in optimizers. But I mean, I wouldn't use Copazi on some of the models we've done, because you need other tooling. So I mean, for small jobs, parameter fitting with Copazi, yeah, that's fine. For big jobs, and if you're doing lots of them, you need to think of something else. So.
Mm.
But I use it every now and then just to check, you know, we get the same results as they do. So
yeah, it can be anytime you have sort of a weird edge case for something, it'd be like, am I actually doing what I think I'm doing? And
yeah.
Michael says reproducibility is awesome, which is great, given that my next talk will be about that. There's a quick exercise. I'm pretty sure everyone can do it. It was just to, be able to load a different model with the, load a different model directly from the Biomodels database. We can give time to work on that, or we can take a short break.
Or both.
Or both. Maybe we can just, it's a very quick exercise. Let's simultaneously take a 10-minute break, and then also if you want to do the exercise, and we can reconvene and go through it. It'll be quite quick.
Okay.
Solve the exercise and want to tell what they did. Otherwise, I can go through it. Okay, we'll go through it together. Okay, so the goal of the exercise was to be able to download this Kolodenko 2000 model of ultrasensitivity and negative feedback oscillations. I just clicked on that link to open it here. And if we go to this page in Biomodels, we see that it has this model ID. This is Biomodel 10. So that's pretty much the majority of the information we need if we have our code from above. So let's just take a look at this cell. So here we're going to do the te.load sbml model, or as people pointed out in the chat, you could also do te.load s, and then provide the URL to the download for this model here. I use the code from above, but just I'm changing this to Biomodel 10. So basically I just substituted this biomodel ID from the code above here and also in the file name here.
I have one quick comment about that.
Yeah.
Just that biomodels, especially some of the later files, they change their naming scheme. So the file name won't necessarily be the same as the model ID _url.xml. It will sometimes change. It'll be like, I don't know, the name of the author.xml or something like that. So you do, especially for any biomodel after, I don't know, 700 or so, you might have to go to the actual files page, look at and get the URL, or at least the name of the file from the actual thing. It's a good rule. Go ahead.
Yeah. Sorry, I just wasn't sure if I was sharing before.
Oh yeah, you were sharing. Yeah.
Okay, my controls looked strange. Okay, great.
I guess up to 700, this could be used as a quick way to download the entire Biomodels database just by incrementing the number.
Yeah, there's actually, I have code to download the entire Biomodels database that actually uses their REST API thing. But yes, this will also work pretty well. So
okay.
yeah.
This is a poor person's way of doing it.
Right.
Yep, so that is that exercise. We'll move into the reproducibility talk. And for that, again, I'll be using this notebook here with the Biomodel 12 ID in the title, and then also just this slide deck. And then this is populated with a lot of resources. So at the very end, I'll just go through a couple of those. And before I start, I just want to say that in the second half of the session today, we'll have Dr. Saro talk a little bit more about some advanced simulation topics, and then we'll leave some time for open questions, and just to like work with the instructors on your research project if you have questions. So that'll be coming up after that. Okay. So reproducibility. This is going to be a sort of practical guide to reproducible modeling. I'll basically go through some tips that we recommend when you're trying to make your research reproducible, and then I'll also go through a sort of implementation of some of those tips, and it'll kind of be, hopefully it'll be a good review for a lot of the steps that you've already heard about earlier in the sessions, and get you thinking back to some of those topics. Right now I have this second Colab notebook open, and I just started running these installations and imports. Lucian was so gracious to help me yesterday as there were some version issues. So in that case, I'll just kind of describe some of the setup here if you want to follow along as well. Basically, the steps to making sure this runs are to first run this installations and imports block of code. Then you're going to go down to this second block, and presumably when you run it, you're going to get this error message that Lucian wrote. And essentially you are installing an updated version of matplotlib, but it's not actually getting reloaded then. So the next step before you go forward is to go ahead and restart your runtime. And then ideally when I run this block, hopefully it's good to go. Okay, good, no error message. So in that case, it'll be good to go. So again, run block 1, run block 2, restart runtime, and then run block 2, and you shouldn't get the error message anymore. Sorry for
You don't
that.
have to run block 2 the first time, just run block 1,
Yeah.
restart, and then go from there. So yeah.
True. Okay, so reproducibility. Why is reproducibility in computational biology important? Well, aside from the fact that there have been numerous calls to try to address the lack of reproducibility across scientific disciplines, there's a recent study a couple years ago that systematically went through models in Biomodles Database, evaluated about 450 or so of those, and found that about 50% of them were not actually reproducible, either due to incorrect information or missing information in the published manuscript alongside that model. But in case that percentage of irreproducible modeling results isn't very convincing to you, we have this fun story that we like to tell, the really fun, very sarcastic, fun story. So let's start with actually this tragic anecdote that illustrates some of the core issues that come with reproducibility, particularly in computational modeling. So this story starts out with two researchers and their research groups, but we'll use them to represent all the agents involved here. So researchers David Chandler at UC Berkeley and also Pablo De Benedetti at Princeton. These two groups were both investigating a very similar problem. They were trying to figure out what water molecules looked like when they were supercooled but still in a liquid state. Basically immediately before the water molecules became ice. What did they look like? And Dr. De Benedetti's group found that liquid water could transition between this high density and low density state without freezing. But Dr. Chandler's group found that there was a single state. Since Dr. Chandler had more renown in his field, it was kind of difficult to oppose his findings, and the De Benedetti group very extensively validated their code and findings to try to ensure that they had confidence in what they thought was happening. And ultimately, they requested to see the code from Dr. Chandler's lab. And over the course of 7 years, the debate remained back and forth. Critiques were happening, more investigation. It wasn't until the end of this period that Dr. Chandler's lab finally actually shared their code. And very quickly, the De Benedetti group was able to figure out that there's this discrepancy in the results that ultimately boiled down to an error in a portion of the code that was used by the Chandler group to add some speed advantages to how fast the simulations could be run. But it actually forced the biophysical system to exist essentially at a much higher temperature than it was believed to be in, and this completely changed the molecular behavior. Had they shared their code earlier on, maybe at the beginning of this debate, this could have potentially been identified sooner, spared some reputations, and importantly, maybe a very notable scientific result would have been communicated with confidence to that community. Of course, it's not always actually the case that two groups are going to investigate the exact same problem, which means that there are probably a lot of problems out there that nobody is actually able to identify, that they can't reproduce that study. So if it can't actually be reproduced, how can we actually have confidence in the finding that's reported? And if we don't share our data and code for computational studies, how is anyone going to actually perform reproduction of our work? So hopefully that's motivating enough to you. Sorry, I missed the SpongeBob edit. 7 years later, and then that's when they shared and figured out what was going on. All right, so for this talk, it's a practical guide to reproducible biomodeling. So we'll talk about some of the steps you can take to make your work more reproducible. And then we'll also talk just like briefly about some cultural shifts that might actually help enable reproducible biomodeling as well. So less on the practical implementation side, but more what we can do as a community. All right, so what practical guidelines can we follow to help make our models reproducible? So what I'm going to talk about today is going to be pretty much outlined in this paper where we shared 9 best practices to make your models reproducible. Unfortunately, I won't be able to comprehensively cover all the practical tools. So for the purpose of this talk, I'll give general guidelines and then also make suggestions for standards and tools that are particularly useful within the subcellular modeling community, most of which are going to be used in Python specifically. One thing to consider when we talk about reproducibility is going to be the FAIR principles, which we use as sort of guiding goals in conducting reproducible research. FAIR stands for findability, accessibility, interoperability, and reuse. And these principles were designed to ensure proper management of data, but they also apply to software and to models as well. Ensuring that our software and data can both be found and accessed allows us to inspect published findings. And ultimately, the #1 reason that studies can't be reproduced is that some critical data or software that's used in the study is not shared, and the publication is often insufficient to represent the workflow. And even if it were sufficient, it would take considerable effort to do so many times. So ensuring that data and software is interoperable allows us to build upon previous work by seamlessly integrating model artifacts into our own workflows, and then reusing existing materials is going to save us time, energy, expenses, and it will maximize the utility of that data and software that was created. We simply don't really need to reinvent the wheel if we don't have to. So reproducibility offers a lot of benefits. Just a note that metadata, which can provide identifier information or context about a data set, model, or code, should also follow these principles. Metadata help us understand the model artifacts and identify them as relevant to our purposes or not. So it's important to make sure they're also, they follow the FAIR principles. And one widely adopted tool that you can use to help make sure that your data and code is fair would be GitHub, which again, which Lucian introduced yesterday. It doesn't only serve as version control repository, which gives you a historical record of the changes that are made to your data and code, but it also makes it very easy to share your study or tools with other researchers. So we would recommend using it, and it's commonly used for sharing your full modeling study. Okay, now we can get kind of into the best practices. And for this, I'll try to switch between the slides and the notebook pretty consistently. So just some more setup for the notebook really quickly. I'm going to clone some information from this GitHub repo, which will just make some files I already have saved accessible to me, accessible to us. And if you want more information about this notebook in particular, there's a little bit more in the GitHub repo itself, so you can also refer back to that. Okay, so this is just cloning in my GitHub repo that is going to be useful. And then we're going to import all of our dependencies. Okay, and before we move into this, let's talk a little bit about data aggregation. So data aggregation is the process of collecting data and metadata that you need to inform your model. Again, metadata, which is data that describes your data, could include the conditions in which your experiment was performed, for example, the cell line used, the temperature, microscopy device settings, and it basically is used. It can be used by other researchers to determine if the experimental conditions used for the data provided actually fits the use case for their model. Another type of metadata is called provenance information. It's basically a historical record, and it tells you how the data was produced and how it was manipulated after it was collected. This can basically tell you the lab of origin of the data, whether statistical analyses had been applied, whether only a subset of the original data was retained. And both of these together, metadata about measurements and provenance information, can help us understand what is in a data set. And that allows us to decide if it's relevant to our study or not, and then how to use it if it is. Many of us have at one point or another used Excel for storing data. As a general principle, if you do use Excel, remember you can read it. Remember that if you can read it and understand the data, that makes it useful, but it's more useful if a computer can actually read it. So it's important to consider ways that you can parse your data programmatically, and this will include often reading the metadata. So the best practice here is to, in the process of aggregation and curation of your data, make sure you maintain its metadata and provenance. And one of the tools that we recommend, a standard tool, is the Scientific Evidence and Provenance Information Ontology. It basically is a set of meaningful names for concepts within a given scientific domain, and it provides a description of how those concepts are related to each other. So it outlines a standardized way to communicate provenance information. And then a recommended tool to use for data aggregation is going to be Bioservices, which is a Python package that provides access to many bioinformatics web services, which means it can be used to programmatically search databases for relevant measurements. And we'll just show a very quick example here. So in this modeling study, we're going to be working with the repressilator again, which you saw introed in day one. And for this first step of data aggregation, I'm just showing a really simple example of using bioservices to search this KEGG database. So first I'm going to set a database. So basically when I did the import here, I specifically imported, where is the line? Oh, okay. Well, I guess I'm probably excessively importing here because I'm only going to use an example with the KEGG database, but from bioservices, I'm importing all of the packages within that. And one of them is KEGG. So here I'm setting the database to be KEGG, and then I want to retrieve a KEGG entry. Specifically, I want to import what is stored with this KEGG identifier 18476, which is going to be well, we'll see what it's going to be, but it's relevant to the repressilator model, and it's essentially going to be TEDR. So here I'm just calling this TEDR query, and I'm using the database I just set to get what is stored at this identifier, and I'm going to then build a dictionary that I can use to parse that query. So I'm calling that TEDR_dict. It gets database.parse TEDR query, and this is just, the details of this are not super important. You'll probably the syntax will be important to look up, or the commands will be important to look up when you are actually interested in using bioservices. But what this is doing is it's just building a dictionary of relevant information that's stored about this molecule with this KEGG ID. So to get a little bit of that information from the dictionary, we're going to search using the key name, so we can figure out what is the name of the species that's stored at this identifier. And then also we're going to search for bright info. This is another it's a sort of hierarchy that can be used to define how species are interrelated. And so we're going to print both of those out, and then, sorry, I should probably run this code. And then we're going to also want to store some of that metadata and experimental measurements. So that's going to happen in the second half of the block, where I'm setting the biomodel ID metadata to be a Panda data frame. And then I think Joe talked a little bit about data frames, but what I'm going to include in it will be the information stored in my dictionary about the name, the information stored about the bright hierarchy, and I'm going to basically name each of those entries with biochemical species name and bright. And then I want to export this data because we want to save it somewhere so that it's retained as information that was collected during our modeling study. So I'm exporting it to Excel, and I'm providing it the file name there. So that's going to happen. That's going to basically, here I'm printing out the name of the species, tetracycline family transcriptional regulator, or tetracycline repressor protein. I know we're familiar with that as being one of the three proteins that is performing this cascade of repression in the repressilator model. And then also this KEGG orthology information contained in, if we query for Bright. So this just gives us a little more information about actually what the tetracycline TEDR protein is. Yep, so both of those information is going to get exported here as metadata that we collected. Okay. That's kind of just a simple example of using bioservices for data aggregation. The next step then, or the next recommended best practice, it has to do with the process of model construction. So here, our best practice is to record the model construction process. This rule has to do with basically using ontologies, standardized naming formats, and a common language format where parts of your model can be specified with distinct and identifiable meaning. So there are existing standard formats that help you to organize your model. So for example, SBML helps you specify your model, which can be written again with a number of software tools. And it has distinct regions in the markup language that can be used to specify the species in your biochemical network, the reaction rates that relate the species to one another. It also has a field for identifiers that can be used to precisely define the biochemical species or your numerical rate law, basically using using standard or agreed upon conventions like those that are in the systems biology ontology, which we'll talk about a bit. Standards for metadata files.
Yeah, as I say,
Yeah.
I think it's in general, the metadata files tend to be RDF, and specifically they tend to be RDF XML. You can write RDF in a few different forms. RDF is basically a triple store format, what they call it. You have a subject, an object, and a verb. Like, so you have your entity, and then your type, like is, and then a thing that it is, or something, or something contains something else, or it is a version of something else, or it comes from this other thing. And the third thing in that is almost always some sort of URI, which usually is also a URL. So a URI is just a unique string identifier, globally, like literally the global unique identifier in the world string for a thing. Usually it is also then helpfully a URL that you can go and look at the information about that. So, and then the subject, the thing that it is just the ID in your original file. If you have a metadata file that's referring something else, then that ID would reference, it's like the SBML element with this ID, or the SBML element with this meta ID, or something like that. And there are also ontologies of the verb in the middle, is version of, contains, comes from, those sorts of things. You'll see a lot of that when you import a biomodels into Antimony. You can see all the annotations at the bottom, and that'll sort of give you an idea of generally the sort of thing that they look like.
The next question was about how much metadata to include. I think that kind of falls into Miriam, would you say would be the standard there? Minimal information required in the annotation of models.
Yeah,
I think it's basically.
Again, so Miriam, the first M is just the minimum, right? So that's you need at least this much, but the more the better. The problem is that it just at some point, you're getting into decreasing rewards for the amount of effort you're putting in. It takes a lot of effort to put in any metadata. There's been a lot of there's been a push in at least our community to try to get people to put more metadata in, and you try to push people to at least get to the minimum information. Obviously, if you kept on going and put in more, that would be great. My feeling is, from watching the community, is that the real value of your metadata comes in years later. Like you publish a model, if you annotate it really well and publish it on Biomodels, say, then in 6 years, someone's looking for that. And if you've annotated it well, they will find your model and say like, oh, hey, they already did all this stuff. So that's a hard sell for the amount of effort it takes today. It's like, it'll pay off 6 years later, I hope. Nonetheless, the more the community does this, then the more people are likely to search for those things, and the sort of everyone is sort of buoyed by a community that in general tries to do these things well. Yeah, the immediate incentive is really low. The close incentive is for future you, like you in 3 years, you're like, I know I worked on this. Like, what was I talking about? And the next level out is like future people in your lab. It's like, so-and-so worked on that. I hope they left good notes. It's sort of like commenting code. It's like, it's not immediately useful, but but when it is useful, there's nothing like it.
Yep. I think we'll get into it in a bit, but I'm pretty sure that the repressolator model itself, it's very well annotated, like extremely well annotated, but the species themselves, the names are not necessarily, you can't identify actually what the species are from the names, but the annotations give you the information about what those species are. So without the annotations, you wouldn't have really any biological relevance in the model, I guess. So
on a practical level, in your SBML model, if you can annotate the species, if you can annotate the reactions, if you can annotate the compartments, that's great. If you can go further, and if you are using like particular, like Michaelis-Menten kinetics, you can annotate the actual kinetics also. But species, reactions, compartments, I'd say that's sort of the main thing that is most useful to annotate. And I guess the other thing would be provenance. Like, like if you are pulling in data from a particular source or from a particular paper, then note that somewhere also.
Okay, so some recommended standards for recording your construction process would be Miriam minimum information guidelines, like we talked about. There's also another tool, completely different tool, is SBGN, or standard is SBGN, the Systems Biology Graphical Notation, which allows you to basically lay out networks in a standardized way. So I'll show an example of that. And then for annotation, some recommended tools would be libomixmeta, which will allow you to write RDF. I believe that's, Herbert can confirm or deny, that libomixmeta writes RDF.
Yeah, it does. Yeah.
Okay. Yeah.
She writes it out to a separate file, and then copies it into a Combine archive.
And then SBML utils, which most likely you'll hear about tomorrow from Matthias Koenig, is another alternative tool for annotation, for providing annotations, and it has other utilities as well. So moving on here. Let's see. Okay, I want to go into the visualization. So we'll just do a really quick step here to import the model from the Biomodles database using Tellurium. Like we just talked about in the last session, you can provide the URL to your to the model you want to download from Biomodles database. And then we're going to just run a simulation just to give you an idea of what the model looks like, although you've seen a little bit about the repressolator before. And you're familiar with all this plotting information. So we're going to have this system that has these oscillations, and then we have, these are what I was mentioning about the species names not necessarily being identifiable. You have PX, PY, and PZ, and then X, Y, and Z. And ultimately, these end up being the 3 repressor proteins, and then the mRNA that's used to produce those proteins. So yep, so that gives you a little bit of an idea of the system. So that could be your first check to try to understand the system. Then we want to basically be able to visualize the model network. This can just help you generally to get a sense of how components of your model are interacting with each other if you're able to generate SBGN. So here, just as a quick note, I actually generated the SBGN file using Cell Designer. I imported the SBML from Biomodles Database and then exported an SBGN ML diagram. And this is the code here isn't necessarily the most important. What it's doing is just reading this file, this SBGN file, using this utils package, and then providing basically providing a temporary file that we're going to then use to visualize with render SBGN. And I believe this is all contained within the Sbml utils package. And then image is just showing the image that's generated in this line. So it's just visualizing that. And this gives you an idea of a visual idea of what's happening in your network. And this is just something that could be useful as an additional form of documentation of your model that helps with understanding. So here we have a little bit more information about what the species are that are involved in the model as well. Okay, let's see. Okay, we're not ready to move on to parameter estimation yet, so we'll talk a little bit about annotation, a practical example of annotation. So when you're annotating a model in the antimony format, I'm going to do this in a really simple way. Just a side note that, like I said, this particular model is very well annotated. So the terms that are going to be included in the Excel file that I'm supplying to the model annotator here, those are already actually contained in the model. So I'm not actually adding new information to the model, but just as an example of how this is done, I wanted to include it. So here I'm going to use the model annotator and read this annotations data frame. So this is from the Sbml utils package, which again, you may hear more about tomorrow. So I'm generating a data frame from this file, from this Excel file, which I just wrote manually or curated manually, specifying the file format as Excel, and then I'm just printing out what this data frame actually looks like, so you can see the information that's contained in it and get an idea of what you might incorporate in your annotations. So here, this first column is basically telling you what the parameter is that I'm adding the annotation to. Pattern is referencing what it's actually named in your SBML file in this case. Then SBML type is telling you what kind of object am I annotating that's a part of SBML, and all of these are going to be parameter values, which you'll see here. Species are also considered parameters here. The annotation type, like Lucien was mentioning, this is going to be encoded as RDF. So all these annotations are going to be RDF annotations. This is information that's used in the RDF in generating the RDF. Less important potentially for us, but then these last two columns are going to actually give you the information about the annotation for each of the biochemical species that we want to apply. So here in resource, this is going to be basically in this case all identifiers that give you some information about what the parameter is, and specifically these resources are going to be SBO terms. This is a systems biology ontology, which has a lot of terms that are standardized across the field for referring to different parameters or model components. And all of these are all of the SBO terms here are then specified with some name in this last column, so you can actually understand what the identifier is. So we have some decay constants, Hill coefficients. We're specifying species as ribonucleic acids or polypeptide chains. And all this information helps us really understand what the components of the model are in terms of the biochemical system. So this adds a lot of value to the.
Are those things actually parameters? Or are they species?
These are species.
You in your SBML type, you claim they were parameters, but
that is
But if you made
probably
it
an
by
error
hand, that
on
could
my
explain
part.
it.
Yep, no, that would be my bad then.
Okay.
But yeah, you should probably declare them as species, and maybe I just made a bad annotation, which is
It might not use that information in the creation of the annotation anyway.
I don't believe it does.
Yeah.
So this might be for like understanding more than anything else.
Yeah.
But I think potentially we can print that out later. Okay, so for then actually performing the annotation, we're gonna go ahead and, well, actually, probably in the case of using this in a collab notebook, we may not need to set this. I won't need to set this base directory. I'm not actually going to use it. So I'm just going to get rid of that actually. But we're going to create the SBML report. And to do that, this is all SBML utils functionality. So you don't have to worry too much about the actual code unless you're planning on using it soon, but you'll hear more tomorrow. So we're using this function annotate SBML, providing the source for the model that we're going to annotate. So in this case, I'm pulling it from the MIMB reproducible biomodeling repository that I cloned, and I'm using the XML file that's in there, which is the SBML. And then the annotations path, I'm providing it the access to the Excel file, which I'm also going to pull from that cloned repository. And then finally, the file path that I want to export the annotated version of the model to. So here, this would be this biomodel 12 _ annotated.xml. So that'll be where I store the annotated version of my model. So we can go ahead and run that code. And if I try to load it, let's see if we can. Oops. Okay. I wanted to just try to confirm or deny if those annotations ended up incorrectly modifying it. So here you'll start to see some of the SBO terms that were added as a result of the annotation. Again, a lot of these were already added to the model, so these aren't necessarily new, but this is telling you all the species, and then the type of annotation it's providing is an SBO term, and then it's providing the number or the identifier number of that SBO term. I don't think it's incorporating information here about, I mean, at least in the antimony, it's not modifying anything about the species. And I don't see any annotations here about.
I think all your annotations are SBO terms, weren't they?
Yeah, they're all SBO terms. I just didn't know if the parameter versus species field would modify anything.
Yeah, I think it looks like it didn't care about that.
So hopefully that is all good. Okay, so yeah, that's a intro to modifying some of the annotations. I think I have my let me go to simulation first, because I think that kind of pops up first in the notebook. But another a sort of next step that we use after this would be then to perform our simulation. And for the best practice here, we want to record all of our inputs and methods, our initial conditions, numerical integration algorithms, random number generator algorithms, and seeds. And the recommended standard for simulation is going to be SetML, which we just talked about. And then some tools that you could use would be Tellurium or Copazi. Again, Copazi is very commonly used, so it's worth it to consider if it might be useful to you. But for the simulation, we'll go through, but this is going to be really similar to what I showed you in the last session. So we want to write some simulation experiments in SetML that use phrased.ml again. So we're going to use some familiar code, load our SBML model from Biomodels, export it to this SBML model, SBML file that we can store in our working directory. And then we can get started with writing our phrase.ml string again. So just to review, we're going to have a model ID, the keyword model, and then provide it with the file name for the SBML that we just generated. And again, we'll run a deterministic simulation. As you saw before, that's done with simulate uniform, start time, stop time, number of time points. We'll perform this task of running our deterministic simulation where we have run this experiment on this model. And we're going to then go ahead and plot repressolator dynamics. And what we'll be plotting will be run deterministic sim. So this task, we're using the time variable against the protein X variable here. So that's our phrase.ml string. It's all the same. It's all the same form as we described last time. And we'll go through the same process of generating the phrase.ml, or sorry, generating setml from our phrase.ml string. So here we're going to just print our setml string that's output as after we do the convert string command, and then we'll go ahead, save that to our file and execute the setml as well. So we'll do all that together. So this is just printing our setml here. And here we have our exported setml. This is the result of executing our setml file. And if you maybe recognize it, this is what I showed you in Copazi earlier that we were able to reproduce in a different simulator. So that is our setml. An alternative method, obviously, you can still run your simulation using Tellurium and you'll get the same oscillatory dynamics here. We're just plotting more species in this case. Okay, so that's going to be useful for our next best practice to do a simulation, which is on storing our simulation results. So there's not necessarily accepted standard in systems biology for storing simulation results, although I think this is something that's actively being discussed. But the best practice here is to save your unprocessed simulation results and to be able to share data that's presented in graphs and tables when you have in some way modified your data to present only a subset of it. So a recommended format would be HDF5. This is a little more structured than using something like Excel, and it allows you to add sort of annotations that can describe your data set. And some recommended tools to do this would be NumPy and Pandas. So I'll show an example of doing that. So with the Tellurium simulation result that we just generated here, we can go ahead and save these data, and we'll do that using HDF5. So here we're going to create an HDF5 file that's done using the H5Py package, and writing file. It's going to be stored with this file name. The data set that we want to use, we're creating the data set from, basically we're going to name it, we're going to create a data set that's going to be stored in our H5F file here. So to do that, we use the command create dataset, provide the name we want to use for that data set, and then pass in some data. So here I'm using simulation result, which was just the result of the simulation that I ran using Tellurium. Then something you could do optionally that'll help you understand the data or use the data potentially. You can add some attributes to your data set. So here, that's done with this attrs, dataset.attrs to basically access. We're going to provide, you can use it to add dataset attributes, and here we want to provide, for example, version information, which could be useful if you potentially run into some incompatibilities when you use an updated version of software, or perhaps some software that you use are incompatible with other software. It might be useful just to troubleshoot that information. So here I'm including adding the attribute Biomodles Database ID. So this is going to tell you what model I actually used to run the simulation. So I'm passing in this Biomodel 12 ID, and then maybe I want to name the model system that I'm studying. So here I'm just naming it with a kind of common name repressolator. and then you'll close the file, and that will basically save it to this file name. So if I then want to load and plot some of the information in the data set just to show you what the results of that HDF5 file look like, I can do that by reading the file instead, which is marked by this R here, and we'll read it into data_hf5f, and then we'll basically go ahead and we'll access specifically this data set that we just named. So again, you could potentially add several data sets to this same HDF5 file and give them different names here, and also give them different attributes, and that could all be contained in a single structured file. Here we only added one, but still, if we want to access the data that corresponds to the specific data set that we named, we're going to use this syntax. This will return all of the data, return it to this variable data. And then if we want to visualize those results, we'll do this slicing as you've seen in different sessions. So here we're taking all of the rows in the first column, and then plotting. So that'll be the time column, or you would expect it to be the time column as before. And then also all of the rows and all of the remaining columns, which should be our species, which you're familiar with, and visualizing that. And then also, if we want to just look at the attributes that are contained within a data set, this is a way to cycle through all the different attributes. So for key in this list we're generating, so we're basically querying all of the attributes which are contained in which have these keys. We can then go ahead and print our key value, and then also whatever information is contained in that whatever information is contained about that named key attribute. So again, I should have probably described this earlier, but this is going to be a dictionary. So when you add this version information, this is going to be your key, and this is going to be the value that is associated with that key in the dictionary. I don't know if we've talked much about dictionaries.
Yeah, I was going to say,
so.
I don't think we've mentioned dictionaries yet, but
yeah,
dictionaries are a useful thing in Python.
Yeah.
Or in any programming language. Go ahead.
Yeah, sorry if that is confusing since we didn't talk about that data structure. But essentially you can query, if you have a key, you can query it using this syntax where you have some dictionary. So this attrs is going to be, or dataset.attrs is going to be a dictionary. You can pass in a key with these square brackets, and you pass in the key as a string here, and then that will basically return. You can query that for the value of it. You'll see an example of what it's going to look like down below, but if we run all of this together, here's where we're visualizing our simulation results of the stored data. So this looks just like it did before. And then for this printing of each of the keys and data, you're getting key, a key being modeled or Biomodels Database ID, and then here's the value for that key model system. Here's the value for that key version information, and here's the value is this basically array containing all the version information for these relevant packages in Tellurium. Hopefully that makes a little bit of sense. Probably the specific syntax or specific commands in this HDF5 package would be something you would want to spend some time troubleshooting, but hopefully at least the concept behind it makes sense. So the value in this is just being able to annotate a data set. You can also contain multiple data sets in a single HDF5 file. It's very structured and it's queryable, given that you know the data set names and also the attributes that you're interested in. Any questions on that? Because given that that was a whole new thing, I may have confused people. Okay, well, in that case, then we'll go into parameter estimation. Okay, so for parameter estimation, the best practice here is that when you're estimating parameters, share the estimation algorithm you use and also perform uncertainty quantification when you can. So some of the recommended tools here, the recommended standard would be PE-tab, which is a newer sort of standardized format that's being adopted by the community. There's now a fair number of tools that support it. And then unfortunately, I don't have an example using PE-tab today. And recommended tools then would be SBstoat if you want to get some parameter estimation up and running pretty quickly on simpler models. SBstoat works quickly. It's easy to write. It's fast. PyBioNetFit can be used for larger models. I know we talked about PyPesto. There's a lot of parameter estimation packages out there that you may choose to use. Kind of just depends on your use case and what you're comfortable with. So for the parameter estimation portion here, we'll just get into a couple of the best practices that you may want to use. So here, I'm going to basically do some parameter estimation as we've seen before with some of Joe's lectures. So a lot of this is going to be somewhat familiar. This will probably take a little bit. Okay, that was pretty quick. So the steps here, I'm setting a random seed, which should ensure that I get consistently the same result. And then I want to basically, because the optimization routines are not going to be deterministic, I'm going to use differential evolution there. So first, to do this parameter estimation, I'm loading a synthetic data set that I have stored in this MIMB reproducible biomodeling folder. This is the file name for that. So I'm going to read that, which is also an HDF5 file. So this is just a fake noisy data set that's supposed to approximate normal data. And then I'm going to get the data. So using the same syntax I described before, where I query for a specific data set and take all of the fields there. And then close the file that we're reading from, because we've now stored our data in data. The next step then will be to basically load a model we want to use for optimizing parameters and decide which parameters we want to actually optimize. So in this case, we're still using the repressilator model. So we're loading that here. And then we're generating a parameter dictionary that we can use. So more dictionaries, hopefully not too confusing. This is going to be used for the specific command that is basically being hidden in this optimize parameters function. But it's using the SciPy optimize differential evolution routine. Or it could be lmfit, I'll have to confirm. But both of which you've probably heard a little bit from Joe's talk. Okay, so here we're deciding which parameters we want to optimize. You might want to optimize all the parameters in your model, but likely you'll be optimizing just a subset that you're not able to identify either from prior knowledge or directly from your experimental data. So here I'm going to just choose 4 parameters in the model that I want to try running this optimization on, and tau mRNA, PS_A, PS_0. And this is going to be, again, it's another dictionary, so I'm sorry if hopefully the syntax mostly makes sense, but this is a key, and then I'm going to also give those values. So the values in this case are going to be a range on the parameter value. So the start range, I think this is start range is 1, stop range is 5. Nope, that's not correct. I may need to double check.
Just a quick note on what
oh
a
yeah,
dictionary
sorry.
is. So just like a list, you can find out the elements of the list by their index. A dictionary is a list where the indexes are names, and the order doesn't matter anymore because everything is named. So right now, so right here, biomodels 12 parameters, we have a list of 3 sublists, one like 0.00115, 00115. They're all, it's a list of 3 things that are all the same. And then instead of an index, there's 0, 1, 2, 3, if they have names, N, tau mRNA, PSA, and PS0. So
yeah, so if you wanted to get the parameter specification for N, you would just query the dictionary using the key as a string N. This is basically, so the reason these are all the same, probably in your modeling study you won't. This is basically saying we want to sample for each of these parameter values from the same space of possible values, which is not likely to happen. I'm using this just for the sake of visualization, but that's just like a side note. These are chosen arbitrarily for the sake of demonstration. They're not based on the biological problem. Okay, so once you've specified all of your parameters, you're going to go ahead and choose which species you want to use for fitting synthetic data. So like Joe talked about, you may do some sort of cross-validation where you're only using a subset of data in your optimization routine, and then you're running it multiple times. Here, I'm going to use the proteins. I'm going to select the proteins as the species I want to use for fitting my simulation to synthetic data, and then initialize some parameter estimation object. This is going to look different depending on what package you're using. So just like a note on that. Here, this parameter estimation routine will take a model, which I can just pass in the Roadrunner object instance here that I specified earlier. When I loaded the SBML model, I'm going to give it some data to use for fitting. I'll give it to the dictionary of parameter, the search space for each of the parameters that I want to optimize. And then I'll also select the species that I want to use from the data set. This is written just using some like ad hoc code that I wrote. So ideally you'll use a more standardized format, especially if you use an existing parameter estimation package like SBstoat or PyPesto. So just consider this as, for the time being, as we're generating some sort of a optimized parameter result that's going to be stored in this biomodel 12_PE, and we'll then be able to basically access some or perform some functions. Like here we can this object is going to be a parameter estimation object, and then we'll be able to perform the optimization using this optimize parameters command here. And then we'll also be able to access information about the optimized parameters after we run this routine. So this is going to return optimized parameters when we run it. This is everything you've already seen from Joe. It's the same general procedure, just probably different code. So when you move down to the next block, I want to print the optimized parameter value that I generated, reset concentrations to some initial value, and reset the selections in the model. And then I want to basically simulate the model using the optimized parameters that I just generated. So that's all going to happen in here. And what this is going to do is basically allow me to visualize the fit that I have to my experimental data. So here I'm plotting the data, the experimental data, which is synthetic. And then I'm also going to plot the results of this simulation here. So here we have, like, you can see generally that you get the oscillatory dynamics that we're expecting from the repository given our prior knowledge, and that it's fitting to this experimental data reasonably well. And in addition to that, I just printed out the optimized values. So this is giving you information about what the parameter was. So for N, and then it tells you this value of 2.075. And again, it's giving you the bounds, which were specified in the parameter estimation dictionary that we talked about. And so it gives you all of those parameter values, all of the optimized values after the routine. Okay, so this is just standard parameter estimation. Now the difference being that we loaded our data from an HDF5 file, which is presumably more structured and has maybe some more metadata. Then the next step that we would recommend would be to generate some information about uncertainty quantification. ideally, you will perform many iterations, which we talked about during Joe's sessions, of optimizations if you're using a stochastic algorithm to try to give an idea of the possible range of values that you might get. So here, this is just going to load the synthetic data set each time and then perform multiple iterations using a Monte Carlo simulation where each time you generate a new fit to some experimental data. Here I'm only running 10 iterations. You should probably run thousands of iterations to get a better idea of what the possible sampling space is for your parameter values. And then I'm going to also save those results to some HDF5 file here using the same code, sending it to HDF, providing the name for the file, the key for the data set that I'm, and the key for the data set that I'm exporting. Okay, this will just take, and you can see it's taking a while. It's not going to be the most efficient optimization routine necessarily, but also it's using differential evolution, which is stochastic, so it takes a while longer. This is only 10 iterations, so this kind of gets at the idea we've talked about previously, that it can take, parameter estimation can be a pretty long procedure to run. While it's going, does anyone have any questions about the parameter estimation so far? I think we're still getting into the Monte Carlo is really the best practice. Performing some sort of uncertainty quantification is very useful. I guess I can talk about the next block of code, which will kind of process the data that's going to be generated. So basically what I'm going to do is I'm going to plot what were these lines? That's basically, okay, so that's just the simulation result. That's like a single simulation result using optimized parameters after you've performed a single parameter estimation routine. So basically optimize the parameters one time and was able to generate these optimized values, and then using those optimized values, running a simulation, this is the fit that I get to the data. Does that make sense? Hopefully that makes sense. Okay. Yeah, I can resolve any doubts later too. Okay, so for this next block of code, it's going to basically just be visualizing the results of the Monte Carlo sampling. So here I'm going to use this is a lot of code, but just to talk about generally what's going on. I'm going to read the Monte Carlo data that I just saved to HDF5. Sorry, there's not 100, there's only 10 parameter sets. And I'm then going to create this radar plot, which is basically just going to plot each of the parameters around a circle and the values they take on, and use that to try to show like how families of parameter values behave. I'll talk about that once I have the visual, what I mean by that. And I'm going to do that by identify families by using just k-means clustering, which is just a kind of machine learning algorithm that can identify it. You specify the number of clusters you want to generate, and then it tries to basically separate the data into distinct populations, which is not related to this course. I'm just using this for the sake of visualization. Then this is all plotting information, and then plotting the confidence interval. I'm going to basically calculate the 95th percentile of the data with upper and lower confidence intervals. You'll see they won't be super encouraging on this particular data set with optimizing random values with random ranges, but we're going to specify the upper and lower confidence intervals, and then we'll do some plotting, and I'll show you what that looks like. Okay, so this is the result that we get here. So again, this radar plot is basically showing you each of the parameters that I wanted to optimize And then along each of their axes, it goes from that 0.0001 to 5, and then the actual like dots on each of those lines is going to be the value it took on in a given optimization routine. So when you see them connected to, when you see the lines connected between parameters, that means that that was like a self-consistent set of parameters that was generated when you optimized all 4 parameters at once. And so, and the 2 different colors are what are being identified by the k-means clustering. So it's identifying this blue set of clusters that is considering one sort of family of values, and this orange set of clusters, which is a second family of values. So it's showing you sets that are very similar to each other, but different, but the sets are different. Within the sets, the parameter values are relatively consistent, but between the sets, they're very different and can be identified as 2 distinct sort of sets of parameters. And as a result, we kind of get these terrible confidence intervals that's like are very wide, where the parameters are fairly likely to take any of the values across this across the range specified, and can take very distinct. For example, in this PS0, it can either be close to 0 or it can be close to a value up here. But that said, this is only running 10 iterations. You would need to run many more to get a better idea of what these confidence intervals look like. But just for the purpose of demonstrating Monte Carlo, that is.
Is that why it looks like it's bimodal? Is that what's coming up on the radar plot?
Yep.
Okay.
Essentially, yeah. Some of the parameters are. Let's see if I still have.
I mean, if you did you only did 10 fits, right?
Right, yeah. It's the 10 fits is not going to give you a really reliable idea of what the
You need to do
parameters are going to
thousands
look
of
like.
them, right?
Yeah, so let's see if I can.
How did you resample? What was the way you did? Was that through the bootstrap? Was it, or what was it?
Oh man, sorry. Yeah, it was with a bootstrap.
Right.
Yeah. So let's see if I have it here somewhere. So with 1,000 values, this is what you actually get, or thousands of values.
It's definitely bimodal.
Yeah, so you kind of get this like clear separation of, well, clear again, I just arbitrarily chose to do 2 clusters, but you kind of get these 2 different families of parameter values that are very similar to each other, but very different. So we recommend reporting that if you do this kind of a test, just because kind of it gives you an idea of if someone else wants to run a simulation and you've only provided 1 set of parameter values, you may completely miss that there's this other possible set, and you may not know for sure what the actual values of those parameters are. So this just kind of is a very simple example that starts to get at that idea.
So ideally, you'd want to see a single polygon.
And
like
probably
just the orange one.
Yeah.
Because you don't want to see, what does it mean that is bimodal here? I mean,
that means you don't know the value of your parameter.
Yeah, it's worse than that though. It means there's 2 possible solutions. So ideally you want 1 solution, and so you want to see in your radar plot just a single polygon basically.
You could probably delve into why there are 2 different solutions, and my guess is, I mean, this is the repressolator, right? So everything is doing, A is affecting B is affecting C. So I would imagine that the difference is like either A affects B a particular way and B affects C a slightly different way, or they reverse what they're doing, and you sort of go around backwards or something. Like that. If everything is set up in a particular way, you can set them up that way, or you can reverse it, or you can do something else.
I don't know. I mean, it's saying that there are 2 sets of parameters that give you the same trajectories.
Yeah.
I mean, it could be just that there's redundancy in the model itself, and that you could be simplified further, or something.
Could be. Yeah, you could have like particular parameters that are dependent on each other.
Yeah, okay, it's interesting.
Okay. Sorry, my Zoom controls keep kind of getting in the way. Okay, so let's see. Okay, let's go back to the guide really quickly. So the next then kind of step of your modeling workflow would be verification and validation, which broadly, as Joe talked about, they cover any testing that can be applied to your model code and also data to ensure that it's robust and also a good representation of the system. Joe talked about generating unit tests, which is one really good method to check that functionality of the code, as well as the behavior of your represented system can automatically basically be rechecked really quickly. And this can check for human errors that are introduced really late in the modeling workflow and ensure that those are caught before you disseminate your work. And then some recommended tools, which Joe has talked about, would be sbmllint and pytest or unit test for Python that you could use. I'll go kind of quickly through the rest of this because I know we're running short on time, but this is going to be really similar to what Joe showed us yesterday, just running a couple unit tests on the model. So for this particular model, I'm going to make this test suite helper, and I will do 2 checks. One will be for mass balance errors, and this is going to use sbmllint. And then I'll also do a check for whether or not the system has complex eigenvalues, because I would expect an oscillating system to have complex eigenvalues. And then implementing the test suite, I have these 2 tests. So testing for the mass balance, which is just asserting false if the model has mass balance errors, and asserting true if the test has asserting true if the test has complex eigenvalues, which is what we would like. We obviously want this to have no mass balance errors in the case for the mass balance test. So we'll run the setup for those 2 for the test suite, and then we'll do a couple quick checks. So here we're going to just load our model, declare go ahead. And then we're going to name it model, so that this is just going to be used for the sake of the test. The way this test suite is run, it needed to be named model, just because of I'm not directly passing this to the test suite. So we're going to run a simulation with the model, which right now should be error free. So we expect it to have oscillatory dynamics because we know what the repressolator simulation is supposed to look like. And then here we're just generating an output so we can confirm that it is oscillating, and then also running the test suite, which will validate whether or not the model is what we expect. So here it's going to run it's going to run the eigenvalues test and also the mass balance test here. Okay, so we get our output. The model's been analyzed. There's no error found. This is a report from sbmllint, and then it ran 2 tests. This is the time it took, and says it's okay. So it didn't identify an error when it was running the tests. Now we're going to modify our model to introduce an error and see if our tests can actually catch that error. So here we're setting the Hill coefficient parameter n to 0, which is going to ultimately remove the oscillatory dynamics from the model. So that's done with this model.n = 0 code. And then we're going to run a simulation and plot it so that we can see that indeed we lost the oscillatory dynamics just for the sake of this example. And then finally run the test suite again. And this time, when we do that, we see, okay, now our system does not seem to have oscillatory dynamics. That's not what we think our system ought to look like. There's no error found in the mass balance. However, it seems like we did fail a test for complex eigenvalues. So it ran 2 tests and it had 1 failure. So we can see that something we've done has introduced an error in our code. Then this last step, which we'll kind of generally get to here, the replication stages. A recommendation we have would be to confirm that your model can be reproduced in an independent computing environment. For example, you might want to use a different simulator to demonstrate that you can generate the same simulation. One recommended tool for that would be biosimulators, and the recommended standard is the Combine archive, which we talked about. And biosimulators actually specifically support the Combine archive. So if you're able to submit if you're able to export your model in the standardized combined format, you'll be able to upload it to a biosimulator, which I think Jonathan will talk about tomorrow, Jonathan Carr, and can check that your simulation can also be run in one of those Docker containers. And finally, another best practice is to package your artifacts, for packaging artifacts and documentation. The best practice is to create packages that contain all of the model artifacts and documentation and deposit those in public version controlled repositories. So again, we talked a little bit about this, but the recommended standard here is the Combine Archive, because it's a standardized package that contains all the necessary materials for regenerating modeling simulations and the model itself. The recommended tool would be biosimulators, again, which can be used to basically provide the whole environment. You could also potentially publish your modeling study to GitHub, as we've talked about, and you can also publish the model to a recommended database like Biomodels or the Fairdom Hub, both of which try to ensure that standards are used and that your model is reproducible. Okay, so the final stage here was just generating that Combine Archive, which we did in the last example, so I won't go through that again. But as I showed you, we were able to reproduce the simulation in Copazi. So that's just one check that it's fairly robust to different simulation environments. To really quickly just go through big picture practical ideas. Generally, the use of standards is going to improve the ease of reproducibility. It helps us to be able to exchange across software, to use software that has good support that we can rely on, and then also have others build off our code. Additionally, self-contained solutions can help us with replication. So if you're able to provide all of the software version information, if you're able to provide the simulator itself, and all the data and the model used in your project, you're going to ensure that somebody else can take all of that information and replicate your results. Model versioning supports iterative development. So we talked a little bit about how GitHub can be helpful. And if you're able to basically build a model that you have reasonable confidence in, run all your analyses on it, and you publish it, that doesn't necessarily have to be the end of the story. You can use that model. You can continue to add to it to make it better. You can always add annotations. You can always include more data that informs the model, and that just makes it stronger and gives us more confidence that it's a good representation of the system. And minimal information criteria or checklists can help us disseminate all the relevant information. So these just are kind of guidelines that help us to know what we should actually be publishing. And software engineering principles, for example, like using version control or iterative development, can help us write clean and comprehensible code. So how can we as modelers try to follow these practices and influence the culture? Well, there are some systems at play, for example, academic journals that are starting to try to incentivize reproducibility. So some journals are starting to offer sort of badges that show what level of reproducibility your work reaches. And this is done by independent curation, which could potentially be one incentive for people adding those annotations or working hard to disseminate their code and data. So another useful method, I guess, for ensuring that we all work on encouraging reproducibility would be to support this as a community initiative. So the Center for Reproducible Biomedical Modeling, which is hosting the summer school, tries to disseminate tools and also information about how we can engage in reproducible biomodeling. And the Computational Modeling in Biology Network also does this largely by developing the standards that we've presented in throughout the course, and ensuring that there's a community consensus on what features we need supported and how those should be represented. And then just big picture cultural ideas. Again, the journals can potentially reward data and code sharing. Individuals can use open source software and open access publishing. And then communities can collaboratively develop tools and standards that provide us both functionality and a common language for communicating our model and results. I just want to acknowledge all those that assisted with this.
A quick addition there, just that as you all are called upon to review papers, if there are models in those papers, you can say, make sure that at least provide the source code for your model, if it's written in R or whatever, and ideally you would provide the SBML version or something like that.
Mm-hmm.
Yeah,
okay.
Yeah, I second that. I mean, it's up to us reviewers to make sure everything is done properly, because the journals are not going to. So.
Okay, that's what I have for today. Unless there are any questions from anyone that I can resolve. Sorry, I ate a little bit into the question time and additional information time. Do we want to take a break?
I think we should take a break.
Oh, wait, sorry. One quick other thing. Sorry. So just in the repo, just some additional tools. I put the paper in case you want like a little bit more information on some of the topics I discussed, but then also we have these checklists that could potentially be helpful just as like an additional tool that you can work through while you're like modeling. It just has some of the steps that you might want to consider doing in each of the different modeling stages. So this one's for if you're using standards specifically. There's also one if you're using general purpose tools or general purpose programming, which is essentially if you're not planning on using standardized formats and you're just writing your own code for a lot of it, not necessarily using a lot of other packages, this one could be helpful. And then hopefully this will be helpful basically if you're looking for information about tools to use for each of these stages. Like I said, I couldn't cover all of them in the presentation, but this table hopefully will give you at least a couple more resources for each of the different stages. You can link out to their documentation if you want. So hopefully that can be used as just as a resource to help you out a little bit. Okay,
Nice.
That's all I've got.
Veronica, were all the files that you were writing out